Okay, I guess I'll go ahead and get started. The sort of roadmap where we're hoping to get to today. Pretty much the whole last class I taught you about drift and issues with respect to estimating effective population size and so on. That provides sort of the table for everything else in biology because all populations are finite in size. Today I want to talk about the evolution of mutation rates, so you'll gain some appreciation of how mutation rates vary across the tree of life. So I don't just work on animals and land plants. We're interested about patterns of evolution across the whole tree of life. And then we'll connect that with specific measures of mutation that we use for quantitative traits that will lead to a situation where we tie mutation and drift together as two simultaneously operating processes and allows us to test certain hypotheses for phenotypic evolution. Then we'll start to link in selection. So I'll be thinking about the joint forces of mutation, selection, and drift all at the same time. And all these cases, I won't be giving you a lot of equations, but I'll be trying to get the essence of the formulae down for you. And then I'm going to try and interlace all these with specific examples that might be useful as a take-home message. And then finally, hopefully I'll get to adding in still one more dimension, recombination, and we'll think about how all these things operate together to define mean phenotype distributions across populations of different sizes, linkage relationships, and so on. And then I hope at the very end I'll get back to this issue of hidden genetic variance that's related to an equation that was over here when Nick finished up yesterday that I think was mentioned. It's a hard way to define things, but hopefully this will just be in terms of biology, biological observations. So I'll start off by reminding you of this thing we call the drift barrier. The idea is pretty simple. Imagine you have some trait has identical features in all species, all phylogenetic lineages under persistent directional selection to increase. It's trying to get up to this thing we might call the level of molecular perfection. That's the limits of biophysics. So an enzyme can't do any better than diffusion limit for substrate molecules and so on. And what's going on here is that selection's always pushing us up in this direction, but as we're going to the right, we're getting to larger and larger effective sizes, selection more and more efficient. And so you're gradually approaching this biophysical limit, but you stall once you get to the point where the strength of selection is balanced by the noise in the evolution process due to drift. So that's the drift barrier. So if you were a researcher with especially long longevity and had the luxury of going out and studying any one of these populations, say, and recording the mean phenotype every 1,000 years for maybe 1 million, 10 million years or so, you would find something like this. You would find that the trait that you're studying is hovering around the drift barrier, but it's wandering around the drift barrier too, because there's transient mutations that allow you to dip down or even to progress above. But in general, you stay in this vicinity. And then there'll be these perhaps bell-shaped distributions. And what you can, they're color-coded here. So we're starting over here at very small effective sizes, larger, larger, and larger. And the variance in the distribution is changing due to the fact we're up in a different location. And then we're finally in a really large population size, but we're still quite a bit from the position of molecular perfection. So that's the basic idea of the drift barrier hypothesis. And I want to apply it to a specific trait, and that is the mutation rate, a trait for which we have a lot of information today, but didn't have so much just a few years ago. So again, to think about this in a more dynamical sense, imagine the trait we're looking at is the error rate in replication. And you might have an allele here, and deleterious mutator alleles could arise, or beneficial anti-mutators could arise. And we're starting with populations in different ancestral states. So up here, we're starting a pretty bad situation. Mutation rate's really, really high. This is bad because most mutations are deleterious. And then selection's able to move the mutation rate gradually down into this gray area, and then you stay there forevermore. And down here, you're starting in a pretty good situation. You have a really low mutation rate, so genomes are really good at faithfully replicating themselves. But the selective advantage here is too small for selection to keep you down there. and you gradually drift back up into the gray area and stay there forevermore. If you start in the gray area, you stay there forevermore. You can see these fluctuations. That's what was giving us those distributions that I showed you before. This isn't purely deterministic. The point here is there's drift around the drift barrier itself. We expect general patterns, but we don't expect everything to be deterministic, as I just said. So we want to think about this now in actual terms of the evolution of the mutation rate. And just to give you an idea how we think selection operates on the mutation rate, let's first think about an asexual population. These are cells in some clonal population, and it's a pretty good state of affairs for the population. Everybody's got 0 or 1 deleterious mutation in it. So it's sort of reached that equilibrium. And then one of the cells picks up some gene that detracts from replication fidelity. So this is a mutator strain. And what that does is it increases the rate of input of genome-wide rate of input of deleterious mutations. And each deleterious mutation is, each deleterious mutation in this example has an effect, a deleterious effect of S. And so that's the rate of removal, in effect, of deleterious mutations from this sublineage. What we want to know then is what is the excess number of deleterious mutations in this lineage. And this is just like a phosphorus budget in a lake. It's, if you know the input rate and the output rate, the ratio there is the excess number of deleterious mutations that you'll eventually get at equilibrium in this particular lineage. So that's the excess number, but we want to know how much pain this mutator is feeling, because that pain is what operates as the selective force for driving the mutation rate down. So we have to multiply the excess number by the effect S, and a long time ago somebody invented numerators and denominators, and that was very nice because we've got one in the denominator, one in the numerator, the S goes away, and the selective disadvantage of a mutator allele in an asexual population turns out to simply be equal to the increase in the genome-wide deleterious mutation rates independent of the effects. Because if the effect of these individual mutations go up, their frequency goes down, and those two things balance out. And so this is expected to be, eventually, to be a pretty small advantage. Once you're up near the drift barrier, as we'll see, mutation rates are already very, very low, and you can't improve things, you can't do any better for the next anti-mutator allele than the difference between 0 and the current rate. So this is likely to be a pretty small number, and that's why it should be influenced by random genetic drift eventually. Now, that's for a asexual population. Things are a bit different for sexual populations. And I'm just imagining here that everything's freely recombining, so it's not that much better normally, but just to give you an idea what's going on. So this is the same cartoon I had before. We've got a mutator arising. And by the way, the selective advantage of an anti-mutator is just the opposite sign is that for a mutator. What's going on here is this mutator segments chromosome into mutators, picking up excess mutation load, but this is a sexual population. So the good news for this guy here is it's exporting a good number of its mutations when it goes through a generation of sex, and they get moved into other cells. If everything's freely recombining, the rate of loss of these just by segregation is, remember, we're thinking about the linkage between the deleterious mutations and the mutator allele itself. And so with free recombination, you lose half the association each generation. And so now up here, we don't have an S in the denominator, we have a 1/2 in the denominator instead. And now when we multiply by S, the S doesn't go away, and that's the selective disadvantage of a mutator. If you, in the extreme case, everything's freely recombining away from the mutator. And there's things in between, of course, when things aren't completely unlinked like this, which they won't normally be. That's the basic theory for trying to understand how selection operates on mutator and anti-mutator alleles. So that's the theory, and we want to start thinking about then how this fits with the data. And 35 years, what year are we in, 2024? 33 years ago, right? Is that correct? Jan Drake wrote this paper in PNAS summarizing everything we knew about mutation rates. And there it all is. There's only 3 cellular organisms here. 3 of these are bacteriophage. And these were based on reporter constructs on top of things, but Jan did some really interesting things with the data and tried to use these indirect measures to estimate the mutation rate per nucleotide site. And then he plotted these as a function of the genome size in these bugs, and he got a slope of -1. And so this is a log-log plot, and he has a slope of -1. So that means the product of the number of nucleotides in the genome and the mutation rate per nucleotide site is a constant. That constant we started calling a Drake, and that is 0.001. So what he is basically saying is about 3 in every 1,000 baby microbes has one new deleterious mutation, if everything fits in this world like this. And he has a great, one of my favorite quotes of all time, he has this great quote in his abstract: because this rate is uniform in such diverse organisms, it's likely to be determined by deep general forces. And I'd like to think those deep general forces are nothing but basic principles of population genetics, as we'll see. So we can do a lot more than what Jan was able to do. I mean, it was a heroic effort at the time, but we've moved into the genomic era, and so now we can do a much better job at estimating mutation rates. We do this by doing things we call mutation accumulation experiments. These are really conceptually simple experiments, but they're quite laborious. It's just an example where this is an experiment. These are paramecium cells, and they reproduce asexually. So we take one mom and feed her really well for a few days, and we're able to then set up, say, 50 replicate lines. They're all starting out genetically identical. Mutation rate's low enough, so in one generation, there's almost, as we'll see, especially in ciliates, these are essentially genetically identical. And then we take every one of these lines every generation or every day and put it through a bottleneck of one cell. And so that shuts down natural selection. The only mutations that are eliminated in experiments like this are ones that are extremely deleterious, which never contribute to anything going on in nature. So lethals, of course, would be eliminated. And then we just do this day after day, week after week, month after month, sometimes year after year. And at the end of the experiment now, we just isolate the lines, and we isolate the DNA from each line, and we sequence the whole genomes. They all started out identical. We know how many generations we went through. We can count the nucleotides we sequenced, and you've got everything you need. You get the complete mutational spectrum, the whole base spectrum for single base substitutions, indel, the spectrum of insertion, deletion sizes, and so on. Well, we usually have backup lines, so usually it's not too much line loss. Technician loss is the bigger thing. But yeah, most things we can store away frozen too. Well, you can freeze anything, of course, but we can thaw them out in a viable fashion. So that's the basic procedure. And this is somewhat of an old table. We've now done about 40 different bacterial species. We're trying to figure out the general rules of the evolution of mutation across the tree of life. These have various different genome sizes, GC contents, and so on. We've now done a couple of archaebacteria. Some of you may know there's this two-domain of life model now, where the bacteria are over there, the archaea are over here, and we grew out of archaea, so we're derived archaebacteria. And then we've done quite a bit. This is a sample of some of the multicellular species we've done. And these are the latest two victims I don't quite have data for, but we're working on the vulvicales now. That's a colonial chlamydomonas, and then we're getting into some of these giant viruses like the giant Mimi virus. So at the end of these experiments, you get data that you can sort of summarize like this. This is an experiment with a soil bacterium, Bacillus subtilis. It's got a genome size of about 4 million bases, and we had 50 lines. We let them go for 5,000 cell divisions. We kind of estimate where the experiment will be using the Drake as guidance. It sort of tells us roughly how many mutations we might expect after 5,000 generations. We're trying to typically collect 3 to 4 to 500 new mutations, so we can really get the spectrum. And so this is, bacteria for the most part have circular genomes, so there's just 50 circles here, and all those dots give you an idea where the mutations are. There's not much of a pattern across the genome in bacteria. One of my colleagues and one of my students would argue a little bit against that. I would agree with them. But when there is a pattern, it's usually very, very weak across the genome, spatially. This is the smallest thing that we know how to grow on a plate, the smallest genome we can grow on a plate. This is a thing called Mesoplasma florum. It was, there's nothing known about this bug other than it was isolated off a flower or a petal of a flower of a lemon tree in Florida. But now we know its mutation rate. So it's got a really tiny genome, less than 1 million bases. We stopped this experiment early because it appeared that the cells were getting sick. In fact, they were. And it's got a mutation. So over here, the bacillus rate is 3 times 10 to the minus 10 per nucleotide site per generation. That's why we have to do so many lines and take them through so many generations. The mutation rate's really low. For Bacillus subtilis, it's about 10 to the minus 8 per nucleotide site per generation. To give you an idea how high that is, the human mutation rate is about 10 to the minus 8 per base per human generation. So these guys are really mutating at warp speed. So that's how we go about and get these data. Yeah. Mitochondrial. Yeah. Oh, they're very different in almost everything. It's hard for us to do that though because mitochondrial genomes are so small. We don't have enough material in this time span. But a few things we've estimated them, and other people have estimated mitochondrial rates by different means. And in animals, they're almost always higher, about 10x higher. Land plants, for reasons we don't know, understand, they're often 10 times lower than the nuclear rates. It's one of the biggest mysteries in mutation rate evolution is why land plant organelle rates. Pardon? The chloroplasts also tend to be low. But there are some plants that have picked up a plant-like mutator allele and they have elevated rates. But those rates are more like animal rates. It's not like they're off the charts.
So what about
Paramecium have, well, I'll be showing you some data. Ciliates have the lowest known mutation rates around, down almost as low as 10 to the minus 12. So those experiments went for a long time and still we only got like 3 dozen mutations or something. It's possible, yeah. Ciliates are reproducing clonally and there's some interesting biology in ciliates. They have expanded nuclei, but these are the germline nuclear mutation rates. So it'd be, just like in our germline. Okay, so we can return to Drake. Remember, that's what Jan would have predicted. Now we have a lot of data for microbes, and we can ask how close do the data fit the expected slope of -1. And well, they're running in the right direction. That's the slope of -1 here. The dark purple ones, that's these guys here. These are unicellular eukaryotes. And then all the blue ones, and there's a couple black dots for archaea, all the blue ones are bacteria. And so the slope for prokaryotes is actually closer to -2, not -1. But it's clear that as bacteria get larger genomes, they do, on average, have lower mutation rates. But keep in mind, there's some noise around these regressions, but that's the general pattern. Looks really good to this point. It's not quantitatively identical what Jan would predict, but still it's running in the right direction. But things change when you start to look at the rates in multicellular species. They're not even running in the expected direction. And so we now have data for a lot of invertebrates, a lot of vascular plants. Arabidopsis, of course, is in there. And a lot of vertebrates now. One thing I failed to mention is, well, of course, we really know the human mutation rate. As I said, it's about 10 to the minus 8. That's not because we do mutation accumulation experiments on humans. And we have a lot of vertebrate rates now because vertebrate mutation rates are high enough. So often one can do what are called trios. You just take mom and dad, sequence their genomes, and then look at genomes in one or more offspring. And typically there's enough mutations in there that in the kids that aren't in the parents. That's sort of like a mini mutation accumulation experiment. So that's why we're getting a lot of good vertebrate estimates these days. So there's no reason to, this is sort of disheartening, the Drake's law doesn't broaden beyond prokaryotes, but there's no reason to think the mutation rate per se should be somehow mystically governed by the genome size. What we're thinking instead, that it would be inversely related to what we call the effective population size. for reasons associated with that drift barrier argument I just made. And so now when you plot, instead of having the genome size down here on the x-axis, we put the effective population size, and now everything's running in the same direction at least. So things with larger effective population sizes tend to have smaller mutation rates, and these guys down here would be ciliates, and that's one filamentous fungus. I've forgotten what it is. So the important point here is the mutation rate per generation varies by about 4 orders of magnitude across the tree of life. So you can't, if we want to do theory in some things, we got to be careful about what mutation rates we apply and think about what organisms the results are applying to. So it scales negatively in every phylogenetic group, no matter what you look at, it's always going negative. I do want to emphasize this is about a 10-fold range of variation at any one effective population size. Some of that's just going to be due to, estimation error. We didn't let our populations go long enough to really refine the rate. Other is because there's genetic variation in the mutation rate within populations, and usually we're only doing this on one genotype per species. And the other thing I wanted to emphasize here is this is one of the only things that I'm aware of that unicellular eukaryotes do better than prokaryotes, and that's replication fidelity. So I don't know if you can see the colors here. The blue dots up here are bacteria, and the purple dots down here are unicellular eukaryotes. And what you can see is that for unicellular eukaryotes, the same effective population size as bacteria, typically the mutation rate per base is reduced by about 10-fold. These are effective population sizes based on heterozygosity. Yeah. So they're what I guess Daniel would call the coalescent effective size.
Mike, one thing to think about for the eukaryote is they have multiple organs of replication, which is a signal for bacteria. So they can probably go slower as they replicate, or individual replicate. That may give error a chance to get corrected.
In principle, but progression forks are really not that different in prokaryotes versus eukaryotes. I think I know what you might be thinking about. I'll get to it. Are you thinking about an artifact? Maybe you're not thinking about the same thing. Yeah, that's right. Yeah, so people don't like that, and I don't like it, but this is the only way you can do it. But you can, there's a lot of ways to think about it. If there's no error in either of those, everything's perfect, right? There's no statistical bias. But a simple way you can check the validity of this is just instead of plotting the mutation rate versus the effective population size, you just plot it against pi. And if things are independent, then you would expect there to be a slope of 1, right? If any is independent of you, and you don't get that. And we've played around with the data to figure out how big the noise would have to be in these data points to give you that kind of statistical artifact. And if you're just dealing within a narrow cloud of mammals, what you're thinking, probably nobody else knows what we're talking about with the exception of a couple people in the room, but unless your range of variation here is really small, this isn't going to cause this negative correlation. We can talk about it more.
Another way to look at it is to say that if you look at pi before you use, then that's how it comes.
Yeah, that's another way of looking at it. I think some people call this Lewontin's paradox, is why is pi so constant? And this actually brings up a good point. This provides a good explanation, a solution to Lewontin's paradox. Because when NE goes up, U goes down, and that keeps the product roughly constant.
Because Drake's law was substitution rate versus the genome size. In a plot,
Drake's plot was genome size down here, yeah.
so they plot NE versus L.
NE versus L. L being genome
Genome
size?
size.
Well,
But you said that
as this goes up, L goes up. So they're going to be L and, but these are rates per site, not per genome. In fact, I'm going to turn it into per genome in the next slide.
I'm just trying to see that can we interpret Drake's observations also combining these?
Yeah, let me go to the next slide. Maybe this is what you're looking for. Because I think this also explains the difference between bacteria and prokaryotes. One thing, sorry, prokaryotes and unicellular eukaryotes here. One of the differences here is that unicellular eukaryotes typically have 3 to 5 times more genes in the genome. So there's more mutational targets. So there are more ways to enforce selection in a downward direction on the mutation rate. And so one way that I broaden, I think this is answering your question. We'll see. What I've done now is I've kept the x-axis constant, and now we're multiplying the mutation rate per site by the number of functional sites in the genome. And so the y-axis now becomes the total number of deleterious mutation rates arising per genome. And you can argue, well, how do you know what sites are under selection and so on? So this is just multiplying by the coding DNA. It really doesn't matter down here in these microbes because 95% or more of the genome is coding DNA. Up here, it might matter, and so you could argue that perhaps 10% or even 20%, if you like, of the genome should be added in, and that's only going to push these values up. And so that's only going to improve the overall fit here. These slopes here are -1. They're just there for guidance to give you an idea of what the slope is here. The slope's a bit less than -1, but not significantly so. And there's a couple points I wanted to emphasize here. I would argue now that replication fidelity is the only trait we have a general theory for across the tree of life. I'm happy for people to criticize this because I'm getting a little nervous. The more and more data we add every year, the fit gets better and better. So this, in my opinion, this fits the drift barrier hypothesis quite well. Again, though, there's an order of magnitude noise around here, around the diagonal axis. Physicists often like to say there's trade-offs between speed and efficiency. And these data don't fit a speed versus efficiency argument because these are the fastest guys down here replicating at the highest rates per unit time, and they have the lowest mutation rates. And then finally, I wanted to point out where we are up here, and we is one of these data points here, being in the vicinity of 1 deleterious mutation rate per genome. That's close to the limit that life can bear based on our ability to purge deleterious mutations. Most of them have small effects, and if the rate at which they're coming in is much greater than natural selection can remove them, you'd be on the road to a mutational meltdown eventually. This is the sum of all mutations in the protein coding genes, non and synonymous. Yeah. In bacteria, as I said, it's essentially the whole genome. So, pardon? Yeah, that's the problem. But what I'm saying is that we could argue all day long about what it's doing down here, but it's hardly going to change anything because we've included essentially the whole genome down here. But it becomes a bigger question up here in invertebrates. And that's why I was saying, I mean, my view, different people might have different views, but I think in a typical mammalian genome, not more than 20% of the genome is regulatory. So you could bounce those points up if you liked, about 20% or so, to just give it a rough feeling of how that would change. That's only improved the fit to a slope of -1. One thing I want to emphasize here is this -1 slope. The data are getting to look pretty good, and people may have alternative explanations. I'd be happy to hear alternative explanations for these patterns. People sometimes interpret the drift barrier hypothesis as implying the slope is going to be -1, and that's not correct. The slope could be almost anything. It depends on the distribution of associated fitness effects and how that changes across here. For the mutation rate, it turns out that it's about -1. If anybody has any ideas from first principles why that should be, I'd be very interested in hearing that. But that's where the data fall out. So the bottom line is we know a lot more about mutation rates than when Jain wrote his paper 30-some years ago. So that's what you need to keep in mind in terms of rate of input of new variation. There's another nice set of observations that I think fit very well the drift vary hypothesis. So in different organisms, there turn out to be multiple DNA polymerases. So E. coli, for example, there's 5, and yeast, there's 5, and humans, we've, I guess, discovered 12 polymerases so far. And some of these are highly error-prone. There's one, you don't want this replicating your genome. Its error rate's like 1 in 10 misincorporations of a nucleotide. These polymerases are important because some of them are built for plowing through big problems on the DNA that there's no way you could get through and replicate the genome. But in doing so, they repair the DNA. You don't have broken DNA, but you replace it with a base that's erroneous. And a number of people have argued, well, these error-prone polymerases have been selected to be error-prone because you want to make a whole lot of mistakes during times of stress. So often these guys here, Pol4 and 5 in bacteria, are listed during times of stress.
Well, conversely, if you're replicating the region of repeated DNA, being error prone means you knock out active normal elements, which could be possible.
Yeah, I'm not sure it would work at that level for a stretch of DNA that big on a single nucleotide. Usually these aren't going through, these are usually only going through a handful of bases at a time. So I would argue that selection hasn't encouraged these error rates, but it's hard for selection to keep those error rates down because these polymerases are only used in a very, very minor fraction of nucleotide transactions in the cell. And so there's not that kind of reinforcement that you get for like delta and epsilon, which replicate almost the entire genome. So I think it's, where's gamma? Who's Greek in the audience? Which one's gamma? Lambda, beta, theta. Oh yeah, I think that, somebody better look this up. I think that might be the mitochondrial. I'm not 100% sure anymore. Okay, so the point is, you know, it's sort of a use it or lose it situation. If you're not really constantly being reinforced, your error rate is expected to drift up over time. They're still really good at carrying out the one function, like plowing through a really bad nucleotide. You know, that's the business end of the molecule. So those are the rates of mutation per site, but this course is a lot about quantitative genetics, is my understanding. And so we now need to think about how you measure mutation rates for quantitative trait variation. And so we have to change our definition now because now this is what we call the mutational variance. It is a function, these are the mutation rates I just talked about, but this would be for nucleotide site for the trait we're interested in. It's a diploid, there's a 2 there. And N is the number of loci contributing to the trait. And we're concerned with phenotypic variation, so we have to multiply it, you know, we're after a variant, so we have to multiply it by the squared effect of the mutation at the phenotypic level. And then this final term here is a dominance, a measure of dominance for the new mutations. And so in the simplest case where the mutations are all additive, this is 0, and this distills down to a simple expression. So U here is the diploid genomic mutation rate, 2n times the rate per site, and we're multiplied by the mean squared effect. So that's the general definition of mutational variance. And we measure the mutational heritability as the ratio of the mutational variance to the environmental variance of the trait. One way to think about that is if you started out with a population with no genetic variance at all, one generation later, that's what the heritability for the trait would be, okay? I'm not one of the folks who believes that we shouldn't use heritability measures. I find these ratios very useful because in a sense, it's a measure of the efficiency of response to selection. How do we do this? We do it exactly the same way. We measure mutational variance exactly the same way we did those molecular experiments. And this is what my lab used to do a lot in the early days before we learned how to sequence DNA. You just run a mutation accumulation experiment again. These happen to be Daphnia. You run them out for a very long period of time. And then these are clones, so you replicate the clones and you measure all the phenotypic traits of interest. You do an analysis of variance, and the between line variance is a measure of the new variation that's arisen by mutation, and then you divide by the number of generations. That's it, okay? So it's a nice link methodologically back to the molecular data. Okay, so here's some numbers. I think it was Nick yesterday who pointed out that heritabilities from mutation accumulation experiments are typically in the range of 10 to the minus 3 to 10 to the minus 2. That was true 30 years ago, and it's still true today, even though there's been more data accumulated, and even things like yeast, people have done this kind of work in, and so on. Why these numbers fall in this particular range of values, I don't know, but that's where they typically fall. I don't think it's a reporting bias, because I think at this time, if somebody got a really gigantic one, we'd be reading about in the literature. And if somebody was really convinced they had a minuscule one, although yeast is pretty low, we'd probably hear about that. So that's where the numbers are. But the way to think about this is, suppose the rate is 0.01. Well, that starting with no variation, that would be the increment of heritability each generation for the first fair number of generations. So after 10 generations, if there was no selection going in, you'd have a heritability of about 10%, and so on. So a mutation for quantitative traits can be a pretty powerful force for introducing new genetic variation. The question is, how much of that variation, of course, is available, is adaptive, available to selection? Well, one set of experiments that's relevant to this is one of the longest term, in terms of absolute time, it is the longest term evolutionary experiment that's been done. This is an experiment that's been done at the University of Illinois, where I started my first job. So I actually got to go out in these fields of maize. So these experiments were started, you remember the starting year, Bruce? 1896. The experiment's still going. Every year, they go out and do exactly the same thing year after year after year. There's only been like 5 investigators involved in this since the beginning. Or the discovery of Mendel, yeah. Yeah, so what's being done in these experiments is there's four sets of lines. This is pathetic looking corn, by the way. This is before hybrid corn days. It's really... Well, not the divergence experiment. Well, okay. Okay, here's time 0, 1896, and then they've been selecting, I forget what it is, I think it's 10% upward and downward for oil content. You can't have corn with less than 0% oil, so we're asymptotically approaching 0. This looks better on a log-log plot. And then they've got 4 other lines for protein, increased protein and decreased protein. It's hard to make it as an organism without protein, so that's never going to go to 0. But what's interesting here, the effective population size I calculated early on, I was going to work on these corn lines because I was really inspired by John Dudley and the work that was going on out there. But then I moved for big trees out in the University of Oregon and never got involved in the experiment directly, but did do some estimates of what they're doing. And the effective population size is about 16. That's summed over pollen grains and female plants. And so the rate of loss of variation should be 1/32 per generation. So after 50 generations or so, there should be almost no original variation left over prior to selection. And yet you can see that's what's happening. You're still getting this linear increase in these lines, and they've done some back selection experiments, and you can move these around a lot. So the most important point here is that this is pretty good evidence of mutational variance arising in these lines over the past 100. It's more than 106 now. There's probably been another 10 years tacked on. A lot of this response to selection today is due to new variation introduced by mutation since the origin of the lines. So one problem with the mutational variance is it's a composite measure, and it doesn't really tell us what the components are, the mutation rate per locus or the average effect. And so people have put a lot of thought into how to get these estimates, and Bateman and Mukai first came up with this clever idea many, many years ago, and they were studying the evolution of evolutionary genetics of viability in fruit flies. And they pointed out, and I'll show you where these equations come from in a second. They pointed out if you did a mutation accumulation experiment with relaxed selection, there will be a directional change in the mean over time. So you can just follow your lines over time and measure the mean. And the slope would be the mutation rate times the average effect. If the average effect is 0, the mean's not going anywhere. So this method's not going to work. You got to have something where the mean is actually changing, as we'll see. But you can estimate that just by regressing the mean on time. And then the rate of change, the lines are going to maybe directionally change over time, but they're also going to diverge over time because they're picking up independent mutations. And then you can measure the rate of increase in the between line variance, and it has that expected value. And what they pointed out was that, well, if you square this value, that slope up here, and multiply by 2, and then divide by this, the 2 goes away, and then you can do a little ranging, and that provides you an estimate of the diploid genomic mutation rate for the trait, divided by this unfortunate term down there. That's 1 plus, that's the coefficient of, the squared coefficient of variation mutational effects. If all your mutations had exactly the same effect, this would be 0, and this would be a great estimator of the mutation rate. But we all agree there's variance in mutational effects at the phenotypic level. So this provides a, let me get to this in a second. And then you can play another trick. If you take half the variance, rate of increase in variance, and divide by the mean, you get an algebraic thing that looks like this. But there is that ugly squared coefficient of variation of mutational effects again. So what this means is that if you play these tricks, you can get because this has got to be greater than equal to 0, then when you play these tricks, you can use this approach here to get a lower bound estimate of the diploid genomic mutation rate, and upper bound estimate of the squared mutational effect. Everybody follow that? It's a pretty clever idea, but there's limitations here. And in fact, there's one other limitation I won't talk about. It's in the book, as we like to say. And these ratios can be biased due to measurement error and so on, but we know how to fix that. Yeah. It doesn't have to change. Yeah, I'm saying if it does, if the mean phenotype doesn't change, you can't use this method. So if you did happen to be at some optimum and there was equal number of mutations going this way and this way,
mutational bias.
Yeah. Well, mutational bias is what will cause this to change, right? So yeah, I think we agree. Okay. The original work that Mukai did, he did some amazingly large experiments with flies, and he was just measuring viability associated with one particular chromosome. And I won't go into, there might be a fly person or two in here that knows Drosophila tricks, but there's neat ways you can pass on certain chromosomes clonally through time with balancer chromosomes. So that's why this method was developed. He was trying to figure out the average, and I'll go through some data on this in a second. Now, this is one approach. Another approach would be to, so this doesn't make any assumptions about the form of the distribution of mutational effects. And because it doesn't make any assumptions, we're stuck with this unknown squared coefficient of variation. But you could make some assumptions. And now you just take all your means and you say, you use maximum likelihood procedures to estimate the mutation rate and the distribution of mutational effects simultaneously. So that's another parameter, or assuming your distribution can be described by 2 parameters. Now you've got to estimate 3 parameters, and you use a maximum likelihood procedure to do this. So Peter Kitely has done a lot of the development in this particular area. Okay, so people have heard about maximum likelihood up to now in different contexts. It's just a way to fit to get the best fit of your parameters with specific assumptions made for the distribution, you get the best fit of the parameters. And you can play around then with different distributions and so on and see if the maximum likelihood changes significantly and so on. Now in principle, I'm not sure anybody's done this yet, but in principle you could condition the whole thing. If you're doing a mutation accumulation experiment where you did the phenotypes and all the molecular work, now you know the genome-wide mutation rate at the nucleotide level as well. That would be an upper bound, right? Your mutation rate for the trait can't be any higher than the molecular rate. So you could at least condition on that, but you'd still need to know something about the fraction of the mutations that are influencing the trait of interest. Turns out a large fraction may be often influencing quantitative traits. So this is going back to the Mukai data. What they concluded was that this is just for viability. The diploid genomic deleterious mutation rate they suggested was at least 0.6 per generation. So they're extrapolating over the whole fly line now, not just one chromosomal segment, but that means that, roughly 60% of newborn flies would have a mutation rate affecting viability. They inferred the average deleterious effect to be about 5%, so the average mutation would reduce viability about 5%, according to these guys. They inferred them to have additive effects. I won't go through how you can do that, but you can do it by, get information by crossing mutation accumulation lines. And how does that fit with everything? Well, we and others have measured the genome-wide base substitution mutation rate, and it's about 1.4 per fly genome per generation. That's the molecular limit here for base substitutions. Some folks have suggested mobile element activity might contribute another 1.2 mutations per generation, a transposon hopping from one site to another. If that seems a little bit high to me, but if it were the case, then the total rate is 2.6 and 0.6 over that. That would imply that over 25% of the new mutations arising in a fly are influencing viability. So maybe we shouldn't be surprised if viability is a giant mutational target. Almost anything you do somehow going to influence the biology. If it's a functional site, is going to influence the biology of the bug.
And the mobile elements.
That might influence the effect size as well, yeah. And when these guys did their experiment, they did not know what the molecular basis of the mutations were. It was done purely statistically using that method I just mentioned. There's been a lot of other experiments, not a lot, but several on C. elegans, some on Arabidopsis, some on yeast, some on Daphnia, and they're more or less concordant. And one of the big surprises is that often up to 20% of the rate for typical quantitative traits, the mutation rate per diploid genome is often at least 20% the genome-wide rate based on molecular data. So, depending on what model you like, you might think of this is like the oligogenic model or something, or the polygenic model. Everything's influencing everything. Omnigenic, I guess is the right word. And often the proportional effects are less than 0.1 per mutation. And typically when people do these experiments, there's one exception done with one experiment in one lab with Arabidopsis, but almost always you find that almost all the lines are getting sicker over time. So there's obviously there has to be a small fraction of beneficial mutations or wouldn't be here, but it's a relatively tiny fraction typically. Yeah, well, there's probably been about 5 to 10 papers written on the fly data. Are there problems with the fly data in some way? Was there line contamination, for example? If you got line contamination, you've got a problem, right? Because you just brought in variation that changed your line mean phenotype. Was mobile element activity unleashed in some way? And Mukai's not around to defend himself, so. Yeah, there's others that are quite high. Yeah. Well, it's much lower in yeast. I've forgotten what it is. Do you remember? It might be right 0.1 or something. Ballpark, yeah. I think the fly are probably the highest rates that are out there. Well, as I said, maybe we shouldn't push this too hard. Problem is, people don't like doing these experiments. It's a lot of work, and the payoff only comes a couple years later, right? So I'm just giving you the data, where they've fallen so far. Yeah, and people looked at that a little bit with Mukai data, and it looks like it bends, but that's all based on one data point. So I'm hesitant to, that was what he was viewing maybe as synergistic epistasis. Yeah, I mean, this is what Mukai said, okay? I'm just telling you what Mukai said. And there's been a lot, it sounds like some of you are aware that there's been a lot of debate about that. But on the other hand, we have data for other species, and I haven't seen that kind of criticism for those data. Okay, so now we're moving on to trying to put two processes together. Now we know about mutation rates, we know about drift, and we want to now think about what happens when mutation and drift operate together. And I'm going to think about this in the context of quantitative traits. So you may remember last class, we talked about at the molecular level, this is the expected equilibrium level of heterozygosity, genetic variation at nucleotide sites under neutrality. So mutations bringing in new variation at rate 2u and pulling it out at rate 1/2ne, and that's the expected standing level of variation. And you can play the same trick for the equilibrium variation for a quantitative trait. So new variation is coming in at rate u now at the diploid level, and we have to multiply it by this. We're interested in phenotypic variation, so we have to multiply it by the average squared value of the effect on the phenotype. That's where that comes in. And that's the same 2ne that we would use up there. And so this is the analog of this nucleotide variation at the level of quantitative traits. Make sense? It's largely just scaling everything over here by the average squared effect, because we're interested in what's going on in the trait, not just counts of mutations at the molecular level. So that's standing within population variance. And then we can think about steady state rate of between species divergence. And we've known a lot about that for a long time at the molecular level. This is one of evolutionary biology's molecular evolutionary biology's laws in the neutral theory of molecular evolution. What is the rate of sequence divergence over time? And if that's the mutation rate and there's n individuals in the population and they're diploid, that's the rate at which the new mutations are coming in. They're neutral, so the probability of fixation is just 1 over the original initial frequency, and the 2n goes away, and the equilibrium rate of divergence is equal to the mutation rate per nucleotide site. Okay? Assuming everything's neutral. And you can do exactly the same thing. This gets back to the question of the directional change in the mean. You can do the same thing for quantitative traits. Now, this is our rate of input of mutations. Again, the big U is at the diploid level. So that's twice the haploid rate. So that's this summed over all sites. That's still the probability of fixation of a new mutation. And then the mutation's becoming fixed. So A is the heterozygous effect, and 2a is the homozygous effect after it becomes fixed. So that's where that 2 comes in, and that becomes our estimate of the rate of change in the mean phenotype. It'll be 0 if the average mutation has no effect, if there's no directional effect, okay? And then finally, we like to think about the rate of increase in the variance among lines, because even if on average they're moving directionally, they can be spreading out over time due to different ones picking up different mutational effects. And that's just twice the mutational variance. So that's the rate of divergence that's the rate at which line mean phenotypes diverge over time. Okay? And I worked out a lot of this when I first discovered quantitative genetics many years ago with Bill Hill. And we wrote this paper in which we suggested that this would be the steady state rate of divergence of a trait. If you went for t generations of divergence between two species, if everything were neutral, that would be the between species variance in the mean phenotype. And that model does make an assumption. It assumes there's essentially unlimited supply of mutations. That you just keep going, you diverge forever. There's noise, but that's the average rate of increase in the between line variance over time. And of course, you can't, for a lot of traits, you're not allowed to go below 0 either, so that's a problem. And then this man here, Clark Cockerham, you can see here, he's looking very stern at us one day, and that's some of our equations, I guess, on the blackboard behind him. Clark Cockerham was one of the founders of modern methodology in quantitative genetics. And he got interested in our paper and said, wait a minute, there must be some limit on mutational effects over time. There's some upper bound to the effect of mutation, some lower bound. And he pointed out that when that happens, then instead of the divergence just going linear over time, it will eventually reach an asymptote, because your two most extreme lines can't go any farther apart than the most extreme mutations. And I would totally agree with this view. But in defense of both methods, you've got to get pretty far out typically before you get into this asymptotic divergence. So for the most part, for not terribly long periods of time, things should be about linear. But the point here is we have some theory. This is neutral theory, makes a minimum number of assumptions. And we can use that theory. People have used this theory to ask various questions in evolutionary genetics. I did this a long time ago. I just realized probably most of the students were not born when this paper was written, but such is life, as they say. This paper was inspired by an interesting idea that Alan Wilson had. He was one of the founders of the field of molecular evolution. He was a very creative guy. And he argued that relative to clams and frogs, mammals evolve at an extraordinarily high rate. And then among mammals, primates evolve really high. And then among primates, the great apes are going really fast. And then we humans are going at warp speed in terms of phenotypic divergence. And his argument was based on an idea he called behavioral drive. And he argued that if you've got a behaviorally sophisticated organism, it can make a behavioral shift to a new environment that its current phenotype works well in. Like I guess if you're a minute bull and you're 7'6" or something, or Christian, you should be in the NBA, I guess, you're pretty tall. Maybe chose the wrong profession, I don't know. If you're short, you become a jockey or something, and that's your family lineage forevermore. You get the basic point. But there's also this interesting side effect of this. He's saying you shift into a new environment, and behaviorally, and that's good, you have a habitat preference, you can stay there, but your phenotype hasn't quite matched what you want in the environment. And then that would somehow propel you into a higher rate of phenotypic evolution. So he called that behavioral drive. And so I thought, well, we can test this with lots known about the phylogeny of mammals, and we ended up getting a lot of bone morphology things for mammals. And then we figured out the rate of divergence of mean phenotypes in these different lineages, and then scaled it and asked, is the rate of divergence rate of increase in between species variance, how does that compare to the mutational variance? Okay, so if things are really being propelled to evolve rapidly, they should be evolving at higher than the neutral rate. And this measure of divergence should exceed our mutational heritability of about 10 to the minus 3. And here we put bounds on it, lower and upper bounds, 10 to the minus 4, 10 to the minus 2. And here's where all the data reside. And what came out of this is everybody in mammals is evolving at rates 1 to 2 to 3 orders of magnitude more slowly than the neutral expectation. So maybe it's justified to say mammals are evolving rapidly at the skeletal level, but they could go a lot faster if they were just phenotypically drifting apart. So another interpretation is that some kind of stabilizing selection is keeping us going as far as we might. And then, on the other hand, us humans are right up there in the neutral range. Okay? So it's just giving you an idea of the kind of things you can do with data and the theory. That's thinking about between line divergence, and then we can think about standing variation in populations. This is a really credible experiment done at the cell biology level on quantitative traits by a guy named Dan Needleman and his PhD student, Reza, here. And Dan first told me he was going to do this experiment. I was at the KATP, and he was just becoming a baby professor at Harvard in the physics department. And he said, I'm going to do this experiment on quantitative genetics and embryos and C. elegans. You're going to be destroyed in the physics department. It sounded really interesting. And he's got tenure that he's doing very, very well. And this is a really neat experiment they did because they're very good microscopists and so on. And they were looking at one particular cell division in C. elegans, and they measured all kinds of traits associated with the mitotic spindle in C. elegans. I think they had something like a dozen traits, including the size of the centrosomes, the position of the division, and so on. And they did this for, I don't know, it was like 40 or 50 different isolates of C. elegans. They measured these traits at the cellular level. And you can see there's a lot of, this is just centrosome size. Remember, centrosomes where the spindle emanates from in a dividing cell. And you can see there's there's about a 2 to 3-fold range of variation in the mean phenotypes. They then estimated that between, you can get the genetic variation in the species of these traits. And so and it turns out that Charlie Baer at the same time was doing a mutation accumulation experiment in C. elegans. Had nothing to do with this, but he had the lines. And so then they measured all these traits in Charlie Baer's lines, and they got the mutational variants for these cell biological traits. And so you've got this within species variance. We know the mutational variants for all these traits, and then you can equate this, divide by this, and you get then you can get an estimate of the effective population size you'd have to account for the standing level of variation. Does it make sense? They're trying to see if this variation, that all of this variation there could be just maintained by neutrality. And if it was maintained by neutrality, then the effective population size of C. elegans would have to be only 20,000. And I think most people agree that the global population of C. elegans' effective size is much bigger than 20,000. So they concluded there must be some kind of perhaps I think they said stabilizing selection on these general cell biological traits. You have to be careful here because many forms of directional selection will also reduce the genetic variance for the trait. But anyhow, they rejected the neutral hypothesis. Maybe not surprising, for a set of traits as key as cell division, but I think it's a nice example of what you can do. All right, so we got up to mutation, drift, and now we're going to add in selection to think about how these three things operate together. And so to do that, we have to think about this thing called the fixation probability of a new mutation. People have talked about it. I don't think we've even introduced the basic formula here yet, but the idea is here, you have a population back here with different allele flavors. That's a new mutation, and due to inheritance just by chance or by selection, something happened, and this red allele goes through and eventually it displaces the yellow and the green. And so all represented in population are the red allele, and that's called a fixation of the red allele. Okay? And so people have spent a lot of time, well, two people in particular, spent some time trying to figure out the probability of fixation of a newly arisen mutation in one copy in the population. It turns out to be a function of the selective advantage. You can use a -s here if it's a selectively disadvantaged mutation. It turns out it's a function of the absolute population size because your original frequency is 1 over twice the population size. And it's also a function of the effective population size. And so that's the famous equation developed by different methods by Malico and by Kimura. It's the probability of fixation of a newly rising mutation with that initial frequency. Okay? And you can plot that out. Is there a question? Okay. You can plot this out. I've plotted it out here under the assumption that n is equal to ne, just to make the graph go easier on you. And so what we're plotting is the probability of fixation. If n is equal to ne, then we can turns out you can plot everything as a function of NES. And that's the probability of fixation of a new mutation. And I just want to focus in on 3 parts of this curve. So if NES, this is the ratio of the strength of selection and the strength of drift, if that's much less than 1, and I'm saying it's in the range of plus or minus 0.1, you're in this range, and your probability of fixation is, the neutral probability of fixation is 1 over 2n. That's the initial frequency. And so if selection's weak, it doesn't matter. You're still going to fix it roughly the neutral rate. That's where you are. If the mutation is strongly deleterious, that is, the product of the effective population size and S is really, really small, you're way over here and you're not going to fix. That should at least be intuitive. If you're highly deleterious, you're never going to spread through the population unless it's minuscule. And then finally, if the mutation is strongly beneficial, now NES is much greater than 1, the probability of fixation turns out to be 2S times the ratio effective and absolute population size. And you'll often hear people say that the probability of fixation of a beneficial allele is just twice its selective advantage. And we get excited about that, or it's nice to point this out to students, which some of you are, that doesn't matter how good you are as a mutation, your probability of fixation is down in the area of twice your selective advantage. So even most beneficial mutations are lost from the population. You can think of some simple reasons for this. There's a beneficial mutation arose in this diploid individual, had a bunch of kids. Oops, none of those kids got the beneficial mutation. They got the alternative allele, and it's gone in 1 generation. And that plays, that game plays out for a number of generations. Oh. Well, I think if you had meiotic drive, it would have to increase the probability of fixation. I mean, in the extreme, suppose your beneficial allele turned everybody to itself. Even then, it doesn't guarantee you're going to go to fixation because none of your progeny might have been promoted.
Yeah,
but
it's additive selection. You get departure from one path. So the drive is fairly small.
okay. Let's see, where was I? Oh, I wanted to emphasize one other aspect of this. So it's not just that the probability of fixation is as small as 2S. It's generally quite a bit smaller than that because this quantity over here, that's the ratio of effective to absolute size. And so in some sense, the ratio of absolute to effective population size is a measure of, it's related to the efficiency of selection. If your NE is really small relative to this probability of fixing it really goes down, okay? So now we want to expand this to think about a simple problem. Let's go back to a 2-allele model, famous big A and little a allele. And both of these alleles are subject to mutation. So little a mutates to big A at that rate, and big A goes to that rate. And one of these alleles might be selectively advantageous. So there might be upward selection pressure or downward selection pressure. And what we're interested in thinking about is if these pressures of mutation and selection remain constant over time, a system like this will reach what's called detailed balance, or sort of a steady state distribution. There'll be a rate at which the rate of these guys going to that and these guys going to that are exactly equal. There's a balance of forces between selection and mutation. And it turns out that everything here in this simple model is a function of one composite parameter. And that composite parameter has 2 parts. This is the mutation pressure it's just the ratio of mutation pressures in 2 directions. So that's what mutation is doing. And this quantity here, this is a really neat result that when you first look at it, you think it's going to be a headache. But if you go back to the Kimura equation and figure out that's the probability of fixation of big A, and then we do the reverse of little a. If you take the ratios and you do about 6 different steps, at least in my hands, everything cancels out. And the ratio of fixation probabilities, that's what this is. It's just a function of 4 times the effective size times S. Okay, so that's the ratio of fixation probabilities, and this is the ratio of mutation, introduction of variation. And you can solve this, and you get this for the steady state probability of the big A allele. And this equation, I believe, was first derived by Winsung Lee, and then later Michael Ballmer derived it. I don't know if anybody did it before them. I don't think so, but I like to call it the Lee-Ballmer equation. And the good news is everything is a function of one composite parameter. The bad news is everything's a function of one composite parameter. So you got to think about the biology underlying all these terms. If everything's neutral, then S is 0 and these terms go away, and the neutral expectation is simply the ratio of the forward mutation rate over the sum of the two. So that's the neutral expectation of being in one or the other state. Now, you can use this formula in an interesting way. Suppose you're looking at some trait and you know the frequency of big A out there, and you happen to know the mutation rates. And you can solve for NES. Okay? That's the only unknown in the equation. And for certain traits, we know what that is. And one of those traits is nucleotide composition in genomes. This is one of the big puzzles in molecular evolution. Yeah, this would be, so this is actually similar to the model you were espousing. I call it the sequential model. So and I'll get back to this in a little bit, but the idea is you're sitting here and then eventually some nice series of events happens and now you're sitting over there for a while and then you go back, okay? So one of the big questions in genome evolution has to do with nucleotide composition in genomes. This is an old graph of mine, just giving you an idea of the distribution of AT contents in genomes across the tree of life. It's actually a little bit broader than that. There's ciliates that have up to 90% A's and T's in their genomes. You know, you need a few G's and C's in there for certain amino acids, and then there's some that get down almost as low as 0.15. So there's this huge distribution. It's not so bad for vertebrates and inverts and land plants, but unicellular eukaryotes are really broad. And then this poses the question, what's driving these differences in nucleotide composition among species? Is it due to some kind of selection pressure? It's good to have A's and T's in certain environments and G's and C's in others. Maybe there's differences in mutation biases in organisms, and that's all it's doing it. And maybe it's not even adaptive, or maybe both. And so we can test this now with data because all these bugs that we have mutation rates in, we also have these mutation rates from GC to AT and AT to GC. We know those in a whole bunch of organisms, and we've sequenced their genomes, so we know the genome composition of A's and T's. So we can solve then for this key parameter, the strength of selection relative to drift. And that's what I'm showing here. So let's just focus on the top panel here. So just to back up, for all these bugs, we first calculate the neutral expectation. That's the nucleotide composition that we'd expect if selection weren't operating. And so this is what we have for a whole bunch of bugs. Some are bacteria, some are unicellular eucs, and some are multicellular eukaryotes. And a couple things I want to point out is that the vast majority of the points are over here. And that's 0.5. And that means that for most species, mutation pressure this is the GC composition. So in most species, mutation pressure is in the AT direction. It's not universal, but most species, that's the prevailing pressure of mutation. So that's the first conclusion. This is based on the total genomic data. And I've broken it up into different components of the genome elsewhere, and we won't talk about that today. But the next point is that the data suggests that it's not just mutation bias, it's not just selection bias, it's both. You can tell that mutation bias is driving things because there is a positive correlation between the genome, a GC composition, and the neutral expectation. So mutation is having an influence, but if you look at the data, you can tell that selection certainly have an influence because almost every point is above the diagonal. So this means there's universal selection essentially for G's and C's over A's and T's. And you can think about why that might be. But that's the universal pattern that one sees. And the final point I want to make down here is that these are the data for silent sites. This is a worry because we use silent sites to estimate theta or 4NEU because we think they're not under selection. But in bacteria, these points are way off the diagonal for a lot of bacteria. So silent sites are clearly under selection in bacterial species. So that's another thing you can do with this theory for simultaneously operating mutation, drift, and selection. Now, we want to add recombination. And I'll go through this fairly quickly because I think I'm supposed to end at the hour, right? Okay, so I'll just remind you that us big eukaryotes, we recombine by crossing over on chromosomes. I showed you this graph before. And this is for bugs and multicellular species across the tree of life. The slope's -1, and there's some vertical variation. But the main point is if you're thinking about the average recombination rate per nucleotide base in organisms, if you just know what the genome size is and the number of chromosomes, you're within about 99% accuracy of the average rate across the genome. There can be variation on chromosomes, but the average is nicely explained by this general one crossover per chromosome arm. I mentioned before, bacteria have rates of recombination in the same vicinity as eukaryotes. This is the ratio of recombination rate to the base rate, mutation rate. And they're all sort of of order 0.1 to 1 or so. And what I want to introduce here is a model to try and understand how you can put all this stuff together to try and understand long-term steady state distributions in populations. So a lot of evolutionary genetics is focused on directional selection. How fast can things go and how far can you get? But there's one can make the argument that a lot of quantitative traits are under persistent directional selection always to move in one direction. And that's the model that I'll be talking about, but this is a general model too. Let's think about the famous, two allele model, two alleles, and to be honest, almost every species, we almost never see any 3 alleles at single nucleotide sites. In our bugs that work on in Daphne, I think 0.1% of sites have 3 alleles. So we're a gift from the gods, as almost everything in biology is 2 alleles. And these are just alternative haplotypes. Everything's additive. So there's only one way to be 4 minuses, one way to be 4 pluses, 4 ways to have a single plus, and 6 ways to have 2 pluses. So there's a combinatorial problem here. This turns out to be important because just because of the multiplicity, it's sort of easy to get driven into this intermediate state. And you can think, what we want to know now is what's the probability of being in these alternative states? These are linkage blocks. There's no recombination going on. And then you can think about the rate of moving from here to here. There's 4 minuses. They're each mutating at that rate. That's the 4, and that's the probability of fixation of a minus to a plus allele. And over here, it's the same expression, but there's a 3 because there's only 3 minuses, 2, only 2 minuses, and so on. And then things can go in the reverse direction. So it's reversible mutation with persistent selection. And we're imagining a trait is under the same selection over evolutionary time. And what we want to think about is what is the steady state distribution over independent lineages of being in these alternative states. This is easy to do if we're back in the sequential model world. You can just solve for this by knowing the value on this and this. The arrow is pointing up and the arrow is pointing down to each state. You can solve this. It works really quite well. But when population sizes are larger and you're not living in that world, it becomes a bit more complicated. Now, I'm going to be focusing on situations where there is a persistent directional selection on a trait, like viability. Presumably, it's usually good to have higher viability. Maybe we don't all agree on that, but I'm all for having enhanced viability. Growth rate, especially in unicellular species, I mean, that's fitness in your particular environment. Traits associated with efficiency of resource utilization, enzyme catalytic rates, DNA replication, and so on. There are many traits that are presumably under stabilizing selection. We don't have a huge number with really good evidence of that, but we love this Gaussian model to use, right? But I think it's fair to say there's traits that are under persistent directional selection as well. In any event, the model that I'm introducing can be used for any kind of fitness function, but today we'll think about directional selection. And so the way to think about this is we're going to have different linkage blocks lengths. And so these would be some stretch of DNA with no recombination in there. And there's 3 flavors of sites. There's 2 alleles at each site. The filled in ball is the good allele and the empty ball is the bad allele. And these would just be 3 haplotypes in the population. And there's 1 optimal nucleotide. The black one's always better than the open one. Okay, and then there's going to be selection on these haplotypes as new mutation is giving rise to variation. And one thing you need to keep in mind here with a model like this, the mutation rates of the haplotypes differ because it's context dependent. Okay, so as one example, the mutation rate for haplotype 1, there's 1 of these guys up here, the big guys, and so that's mutating at that rate, but there's none of these in that stretch. There's 3 of these intermediate beneficials, so that's the rate of these to these, and there's 2 of these, and so on. You get the idea? So every haplotype will have a unique mutation rate, and that must be true in biology in general. And then we're going to use a exponential fitness function, and so these are the selective disadvantages of the bad allele at each site, and then we take them to the power, the number of bad nucleotides in the haplotype block. So just to give you an example, for haplotype 3, there's no bad alleles for the site with large effects, so that's 0, that goes away. There is 1 of these bad ones, and so it's 1 minus S2 to the 1th, and there's 10 of these little white balls, okay? And so that's the fitness of that particular haplotype, and that can be done for all haplotypes. What we're going to think about now is what happens when we increase the selection coefficient, when we increase the degree of linkage, and when we add in different effects. So this is easy to do if there's free recombination. And this was worked out by Kimura, Maruyama, and Crow quite a few years ago. So this is the absolute population size, and we're looking at the frequency of the populations that will be fixed for the beneficial allele. We have replicate populations. We're trying to think of the steady state probability that you're fixed for the good versus the bad allele here. And that's it for mutations with very large effect. That's the view from moderate effects and small effects. Okay, so you get the idea. So as you're way over here with a small population, you can't use these mutations at all. It's not till you get far enough, then this guy goes up and you can take advantage of the big ones. And then as you move across, you can start to take advantage of the moderate ones, and then the really little ones you have to be a really big effective population size. And so we know how to work all that out. And these are just these curves redone. That's the problem. And we can just call that relative growth rate. It's the fraction of nucleotide sites that are fixed for plus. This starts to break down when you've got linkage. So it's just one example. Suppose there's only 5 nucleotide sites, and I've just illustrated 4 ways you could have 3 plus alleles. And there's other ways I think there's other ways you could have 2 white balls and 3 red balls there, okay? This is a multiplicity problem. The problem with this is selection would like to fix the red ball at every one of those sites, but these are completely linked. And so selection can't tell these alternative haplotypes apart. And so this causes selective interference. And so if that's all you do, and you have a certain linkage group length, like 10 to the 4th, 5th, or 6th, it changes these curves to these curves. It makes these shallower. These kinds of things are like drift barriers, right? And what's happening now, though, with linkage, because of the interference that's going on, the gradient becomes more and more shallow. And it becomes especially more shallow with mutations of small effect, okay? So that's one problem with biology. Our nucleotides are linked together, but it's hard to go through meiosis with 3 billion different chromosomes, I guess. So we don't do it that way. These are the kind of plots I just showed you, and it looked to us that they're all sigmoid, and they seem to have the same flavor, the same sort of behavior as the Lee-Balmer equation, which I showed you a few minutes ago. And so we're thinking, well, it's not Lee-Balmer because this doesn't work with the absolute population size. But maybe if we could figure out with a system like this what the effective population size was, we could get everything to work. And we figured that out, and that's why these lines go through the data points from simulation. So what we're trying to come up with is this thing phi, which is a fraction less than 1 multiplying the absolute population size. And we thought, well, let's just figure out the diversity of linked neutral marker and then back calculate NE at the linked neutral marker. That's what we do in nature, right? We estimate NE from polymorphism at neutral markers, but that turns out not to work. And this gets back to the point I was making last lecture I gave, that the coalescent NE is not necessarily NE governing other factors in evolution. And so we eventually worked this out. I worked this out with Archana Devi, who used to be here. She was a PhD student of Kavita Jain's, and she was in our lab for a year. And I won't go through the contortions. There's a lot of math involved here. But this is the magnitude of if this gets lower, it means there's more interference. And you can see there's sort of some cool scaling features that come out of this, like this phi decreases with the cube root. I won't explain where that comes from. But it's of the product of the selected disadvantage, the number of loci, and the mutation rate. Anyhow, we've been able to figure that out. And now let's move on to one further step. All that work was done with mutations of one effect, but we know in nature, different nucleotide sites and so on have different potential for fitness effects. So that previous slide there, that's these plots, the same plots here, but what I've done now is scaled them not as the absolute population size, but the effective size, and they're all bundled together in a nice way. But now what we say is there's a set of sites with large effects like before, but another set with smaller effects. And the way I've got this set up, there's sites that have tenfold lower selective effects, but those are 10 times more frequent. So the moderate effect and the large effect sites have the same effect on the character, but it's divvied up 50-50. And so what happens now as you move to the right, so again, going back here, with constant effects, if you're plotting against any, it's whoosh. The drift barrier, everything's over in one order of magnitude change in population size. If you're too far over here, you're fixed for the bad allele. If you're over here, you're fixed for the good one, but everything happens rapidly. But with two effects, what's going on is the major effect locus is going up, and now essentially they're all fixed, but you're not at a large enough population size quite enough for the moderate effect was be felt by natural selection. And then finally they take over and they move you up. And you see what's happened, it's taken 2 orders of magnitude population size. So the gradient has become shallower. And if you do this with a third effect, like the cartoon I was showing, it would take 3 orders of magnitude. So this is sort of a population genetic perspective on that thing I called the drift barrier before. And the more and more biology you add to this, the more you can get at this problem. So I don't know if I probably should stop now. I've got a little bit. I was going to try and explain biologically a thing that Nick explained mathematically, but we're probably close enough. I should. Okay, I'll do it fast. And I won't do it all, okay? But we work on Daphne. You'll hear more about that next week. And these are really nice. The reason I'm pointing this out, Nick pointed out this problem of linkage and linkage disequilibrium and creating like hidden variation in populations. This probably happens in all populations, but if it's a sexual population, it happens so fast you can't really see it in one generation. But Daphnia have sex our guys have sex every 5 generations. The point I'm going to be making is you can see this buildup. So they're what called cyclical parthogens. They're reproducing sexually. Pond starts to dry up, boom, they all go sexual. Those are boys, those are girls. They make resting eggs and they hatch out the next year. I'm going to skip over this. So this is sort of what Nick was talking about, I believe, and I'm probably using different language, but the idea is there's expressed genetic variance. That's what we measure when we measure heritabilities for traits and so on. But there can be this pool of what we call hidden genetic variation over here that you can't see. It's hidden because of linkage disequilibrium. And if you've got a sexual population, selection's always creating hidden genetic variance, and then you can have sex and recombination. It's converting it back. And so you have the steady state level of both pools. And I think that's what Nick was talking about. Now I'm going to show you a cartoon of how this works. We have a population of some population. Everything's additive. These are chromosomal segments. Everybody adds up to a 12. And I've really exaggerated mutation here. So everything with a red ball is, that's a mutant. would never happen in real population, but I want you to see what's going on. And these are double mutants. And so everybody started out as a 12, and these are still 12 because there was an up mutation and it was balanced by a down mutation. And then there's apparently really strong selection operating on this population. Only 12s are allowed to survive. And so all these guys after selection are 12s, but if you take a closer look, they have different underlying genetic constitutions. So there's linkage, negative linkage disequilibrium. And so this is what we expect to happen if you forego sex. For a long time, this pool of hidden genetic variants will just keep building up and up and up. You'll just be building up more and more negative covariants. And the prediction here is that in an organism that's got periodic sex, you'll get cycles of rates of evolution because you'll have just had about a sex, and now you've got a lot of released hidden genetic variants, you evolve, but now you're hiding it, and then boom, you go up again, next bout of sex, and so on. You get these sort of stepwise changes. That's what the expressed genetic variance is doing, whereas in a sexual species, it's constant. And you can see this in Daphnia. The beauty of this system is you can have a few generations to see the buildup of negative linkage disequilibrium. If you measure heritabilities for traits over one season, these are actually 2 years, you see this downward trend in the heritability. But then when you go back out to the population the next year, whoosh, it's gone right back up. So in this pond, you get a 44% decrease of genetic variation during clonal selection. Then it has sex, and you get this re-release of the hidden genetic variants. This is another example, another lake. These are just individuals brought in the lab. There's no selection. They're brought in, they're carrying resting eggs. We hatch out the resting eggs. We know the genotypes of the mom. And so we can look at the genetic variants in the kids relative to the mom, 20% increase. This one was interesting. The genetic variants actually decreased after sex. That means there's not negative disequilibrium, but positive disequilibrium. And we repeated this and got another similar thing. So how do you get a decrease in variation after sex? Well, this can be explained by positive disequilibrium. So suppose we have these 3 genetic values here. These are diploids. They're in Hardy-Weinberg equilibrium. So that's the genetic variance for the trait, 0.125. And then we bump off the one in the middle. And this is all that's left. And then we let them have sex. So it's 50-50. These chromosome types and these chromotype types are right back to what we were before. So here we have, after selection, the genetic variation builds up, and then it goes down. So you can get these cycles of genetic variation for quantitative traits in populations depending on the form of selection operating on the trait. I think I should stop there. There's more I could talk about, but I've gone over time.